Okay, so thank you for joining me today. What we're going to talk about is basically an overview of how you can use public data sets to basically enhance your research. I'm sure most people here have looked at a paper recently that is going to include some kind of omics level data. And a lot of it is publicly available, and that makes it a huge asset for basically, public data sharing and also for the entire research community. So there's a few reasons that I think using public data is a great resource. So let's start with, I do want to acknowledge, first of all, that the University of Alberta is on Treaty 6 territory, and that we really respect the homeland of the peoples who live here. All right. So why should you care about publicly available data? This is something that I get asked every time I kind of champion this cause. And there's a couple of reasons I think it's important. So first of all, a lot of the time we're reading a paper and we're like, oh, this gene looks interesting, or oh, this study, was interesting, and I wonder if what I'm studying is relevant to this. So a lot of the publicly available data gives you a way of kind of looking at real world patient data to see if your gene is interesting for your research, or is your gene is like relevant to cancer patients. And it's also good to kind of generate ideas, because a lot of with some of the things I'm going to show you, you'll be able to see that if you have a gene of interest, you can look at what else is associated with that gene. And so it can also kind of fuel ideas. One of the other important things that the publicly available data sets can do is it can actually build collaborations. Because if you access someone's data and you use it, it gives you the chance to then approach them in the future if you like, and then say, thank you for your data. I've kind of found this. Maybe we would be interested in collaborating. I want to point out that if you take one of these publicly available data sets and you kind of analyze it, you can absolutely publish this. You can use it in a grant, you can use it in a paper. And as long as you cite the people who generated the data, there is absolutely no problem to you doing this. It's actually really encouraged because it's showing that you're using resources that are available and kind of leveraging what is out there and not duplicating work. Another important thing that I get asked a lot as well, I have to know how to code in order to use a lot of these data. You'll definitely be able to get more out of the data if you can code, but I'm going to show you a lot of resources today that you don't have to know any coding to use. As you'll see, there's a lot of online tools that are being generated today that are amazing. Thank you in advance to the people who have generated those. Another good thing as a PI, it is my duty to point out it's really cheap because it doesn't cost me anything or your PI anything to do this. And it can be really helpful, again, as I said. And another important thing is that, a lot of this work that goes into making these omics data sets is publicly funded. So that's your tax dollars at work, or if you're not paying taxes yet, that's your family's tax dollars at work and your future tax dollars at work. So you may as well make use of these data, given that they are available. So our goals today are a couple. The first is we're going to talk about how we can find data sets that you read about in papers, and then how you can work on analyzing those yourselves. The second thing is going to be learning how to find data sets that are of interest that you can analyze without actually having to, first identify one in a paper. So let's say you research a particular type of cancer, finding a data set that contains relevant information for what you want to use, and then kind of being able to analyze it. And then finally, if you have generated some omics level data, one of the great things about a lot of the tools I'm going to show you today is that you can actually put your own data into these tools and then analyze it in exactly the same way that you're analyzing the public data sets. And you can actually make a really good comparison, kind of apples to apples of both sets of data. So we're going to start today with looking at what is called the gene expression omnibus. And if you've ever read an omics level paper and then kind of scroll down to the methods, what you'll see is often in the methods, they'll say, we've deposited our omics data in the GEO database. So this is what the GEO database looks like. And what I'm going to do today is I'm going to kind of give an overview of each of the tools that I'm talking about, and then I'm going to switch over to the website and I'm going to kind of do a bit of a live demo of the tools so that you can kind of see them in action. And once again, if anybody has questions, just feel free to unmute your microphone and interrupt at any point in time. So what does the GEO database do? So it allows you to download specific data sets that you've seen in papers. You don't have to have a pre-identified data set to use this. You can go onto the GEO data set and just type in a keyword, find what kind of data is available, and then actually analyze it. And one of the great things that they've recently started doing is they've actually put a bunch of analysis tools directly onto this website, which just hugely increases the power of what you can do with the websites. So if you actually go into the database, what you'll see is there's a search box over here. And you can, so right now I've just entered in glioblastoma as an example of something that I was interested in looking at. If you have a GSC number from a paper that you're interested in, you can enter that here. And then it will pull up a bunch of studies that are relevant to whatever search term that you put in. If you click on one of these data sets, what it will do is it will kind of give you a little bit more information about the data set at the bottom. It'll kind of give a quick summary of what the data is. If there's a paper associated with it, it will show the paper. And you can see it will show you just kind of a bit of an overview on the right here about what types of data are available for that particular data set. And then there'll be the reference number on the bottom here. So if you then want to look at this data set, what you can do is click on this particular number, and it's going to pull up a page like this. And what this is basically a record of that data set. So it's going to give you the title of the data set, a summary of what's in there, the contributors. It's going to give you kind of details of who contributed what. The important part's down here. So it's going to give you an idea of all of the samples that are contained in your data set. It's going to give you a link to all of the files. So one of the things about the GEO datasets or geodatasets is that they actually ask users to upload both kind of a process data file and also the raw data files. So you can actually download either the process data files or the raw data files. And that will allow you to either do kind of a really in-depth analysis if you want to use the raw data files, or it will allow you to do kind of a more quick analysis if you pull just the kind of process data files. But the new thing that they've actually put on here, and I will caveat this by saying that this link is not available for all the data sets, but the number of data sets it's available for is increasing really rapidly. So this is called GEO2R, and it's a way to actually analyze each data set online. So I'm going to go over what this analysis will do. And then I'm going to talk about if you find a data set that doesn't have that button, how can you then use it afterwards with some of the other publicly available sets. So if you click on this GEO2R button, it will bring up an example of the study, or I should say details of the study. And what we're looking at here is all of the different samples that were uploaded in the study. So in this particular case, this research contains 2 replicates of each of these treatment conditions or cell lines and treatment conditions. And I can then choose which ones I want to analyze. I can include all of them, or I can choose a subset. So there's a button over here that will allow me to define groups. And in this case, I've selected 2 and I've given them the name of control. I've selected 2 and called them temozolomide, and I've selected 2 and I've called them temozolomide Y15. And then what you can do is once you've done this, you can press on the analysis button. And what it will do is it will run a quick analysis and it will pull up a data page that looks like this. And you'll see that it's going to pull up a bunch of visualizations. And the ones that have a green box around them, if you click on those, they're actually going to bring up the graph, and it's actually going to start an interactive window that you're actually going to be able to look at and do comparisons with. You've also got the full data set down here. So they've put kind of all of the data. And I want to point out that these data are fully downloadable. So you can actually kind of have the analysis done for you in this program and then download the data directly to your computer in something like an Excel file. So it makes it very easy to access. And the other thing I want to point out is that if you've got a bunch of tabs up here, you can actually look at the R script behind it. So you can see exactly how your analysis was done. And that's really important for transparency. And if you ever wanted to report it in a paper, then you can copy the R code and you can say, this is the R code that I used. And that's perfectly legitimate and perfectly fine. So if you then go ahead and click on this data set, so this is a volcano plot that's comparing two different data points. And again, if I pull it up, I end up with this explore and download button. And when I actually click on that, it actually brings me to an interactive window where I can choose which two of my defined control conditions I want to compare. And it allows me to then kind of hover over any one of these data points and see the information. And it also allows me to download the specific genes. And that includes expression data, and it includes significance also. So all of the stats are kind of done for you. So it's a really powerful tool that is very, very versatile. Okay, so we're going to switch over to the website and we're going to do a quick look at what that looks like. All right, so here is the geodata set. And what we want to do now, so I've shared links in the chat button or in the chat that has links to pretty much all of the data sets that we're going to cover today or software. So we can go to Dataset Browser over here, or you can go to the advanced search. And what you'll see is if I do this, then you can see that I've got a bunch of links that have already come up. I'm just going to shift this down. And what you'll see here is if I enter in glioblastoma, I can pull up kind of a bunch of studies that are about glioblastoma. Again, if you've read a paper and want to look at a specific data set, you can put in one of these GSE numbers that you'll see in the paper. And what you see when I do that is I pull up this particular data set. So I can click on this GSE button here, and it will bring me to the web page that I showed you before, where we have the information about the paper, the information about who downloaded it. And then you can see that we've got the different samples that are present here. We've got all of these different data down here that you can download. And again, the raw data files are these ones here. And the nice thing about it is they always tell you how big the file is, so that it's actually reasonable to be able to download without, crazy amounts of storage space. And that's again because they've done all of the processing for you, but they've put the raw material here. So if we want to then analyze this, we can click on this Analyze with the Geo2, and it will bring me to this website where now I can see. So you can see I've got different groups that are present here. And I can either choose to do my analysis with all of these groups, or I can choose a few that are very specific. So in this particular case, I could choose something like, I can go to Define Groups here, and I can say, let's see, so we do not have a temozolomide or a temozolomide group here. So we can say here that I'm going to have a U87, and you can see that I'm going to have a GB4. And you can see, so I've now got different groups here. And the way that you basically tell the program what you want to analyze, oh, we're going to put a GB7 there too. There we go. Okay. So what I can do is I can click on this GB7 line, and I can now click on GB7 here, and you see it's going to color code it according to the group that I selected. I can click on the GB4, and I can say I want this one to be a GB4. And I can click on the U87, which is nicely hidden behind there, and I can say I want this one to be a U87. So you can really easily kind of define your groups. Again, if you wanted to compare all of them, you can absolutely do that. But once you've done that, you can come down here, and it does, by the way, have instructions for what you're supposed to do right here. And then you can click on the Analyze button, and it will run a quick analysis, and then it will pull up the kind of data that I was just showing you. So a lot of these analyses do take a few times. Ooh, what is this saying? Your quest cannot be completed because the R output gives you a hint of what is possible. Interesting. Okay. So let's go back to, oops, there we go. Okay, so I'm going to come back to this one. And okay, so we're going to try a different data set. So sometimes you're going to find some of them that are not going to work. And I could take the time and run through that particular analysis and find the error, but because we are time constrained today, I'm going to not do that. So I'm going to put my temozolomide group back here and my, all right. So we're going to reassign these groups here. So this is going to be the Y15. Temozolomide is going to be here, and then our control groups are up here. All right. So now we can go down here and we can do analyze. And this is the one I ran yesterday, so hopefully it's going to behave itself. And most of the time when you do run into an error like that, it's a pretty quick fix in order to get it to run, or it's something simple that you can go in and tweak. Okay, so here is what you normally get when you run the analyses. And you'll see I've got the list of genes down here, and it gives me p-values in the adjusted p-value, which is the one you really need. But what you really want to do is, this is comparing all three groups that we assign, but most of the time you really want to look at just two groups. So if I go in here into this volcano plot, I can click on explore and download. And then I can actually get the interactive visualization where I can see, you can see I've got my different comparison groups that I can do here. And I can then actually hover over any of these individual bars, and I can get an idea of what is the significance, what is the gene. And again, you can download each of these just in an Excel file that you can then kind of run through Prism or run through a more detailed analysis. So it gives a lot of flexibility in terms of what you can do. There we go. Okay, so we're going to switch back to the PowerPoint, and we're going to talk about what happens if you don't already have a data set identified. And you just basically, you've identified a cool gene and you want to see, or you've read about a cool gene in a paper, and you want to see, is this applicable to my research? Is it related to the gene that I study? How do I go about doing that? So the Genomics Data Commons is, again, a resource available by the NIH, so that's the National Institutes of Health. And what it allows you to do is it allows you to compare information across patients within a data set or between different data sets. And each of these sites that I'm going to talk about is going to contain a different level of information. This particular one is going to allow you to look at mutations in gene status and kind of do correlations with clinical information like survival. You can see when you first go onto the website, it will give you kind of a breakdown of what types of information is there. And it will tell you, there's like 86,962 cases on here, and you've got 68 primary cancer sites, 78 projects. And so there's a lot of data that you can go ahead and access on the site. So if you launch the data portal, what you end up with is a website that looks like this. And it starts off by showing you everything that's on the website, which is not necessarily helpful, but you can really quickly narrow things down. So you can come onto the left over here, and you can quickly narrow into what tissue site you're interested in. And then within there, you can choose either the different databases that are available or the different data sets. You can zoom into a very specific type of cancer. And you can see that there's all the information about the project is present up here. When we look at it in the interactive site in a minute, you'll see kind of what you can do with those data. And there's also a bunch of tools that are available up at the top. So right now we're just looking at kind of the overview. One of the things we can do is we can click on the analysis tab. And this is where we can actually kind of, compare different compare different groups, do an analysis of the clinical statistics, or look at a cohort comparison. So we're going to go over the cohort comparison today, because that's going to be one of the probably most likely things that you're going to want to do. So if we switch back to website, okay, let's quickly scroll through here. So this is the portal, what you get when you do the analysis. And so if I click over here on, let's go to exploration. So this is the page we were just looking at, and it's telling us kind of, how many cases are available for each type of tissue, what type of projects, what type of disease. Most of them are going to be cancer. So I can go in here, and let's say I want to look at breast cancer. So I can click on this particular link here, and each time it's going to narrow down the data that I'm looking at. I can choose a particular data set, or I can choose to include all of them. I can scroll down here. So let's say there's a particular type of data that I want to look at, or a particular type of breast cancer. I can kind of narrow down my search here. Now, let's say I'm interested in looking at like a particular gene or something like that. I can go to the Genes tab here, and now it's going to show me a breakdown of kind of the most common mutations that are present. And then I can come up here and I can search for a particular gene. So let's say we're going to look at the BRCA1 gene because it's one of the most common. So we're going to choose this gene here. And what it's going to do is it's going to give me kind of more specific analysis about each of these. And it's also going to give me the chance to, okay, there we go. So I can then go in and I can choose kind of different genes that are related, and I can look at the different information that's available. I can look at specific mutations if I'm interested in them. And each time you can zoom in and you can actually do kind of the comparison that you want to do. So we're going to switch back now. I realize that the controls for Zoom are very helpfully covering all of the nice tabs that I aligned to help me out here today. So let's see if I can move those. There we go. All right. Okay. So we're going to switch back now to what I think is one of the best tools available. And this is the Cancer Genome Atlas, which is, or you've probably heard it as the TCGA. And this is what is going to give you kind of a huge access to really, really well curated data sets. So what is TCGA? It's basically a collection of very, very thoroughly analyzed cancer data sets that have been made publicly available. So a consortium of researchers from around the world basically decided that they wanted to take a set of cancer samples and to do really deep analysis for all of them. So they've done, for all of the same cases, they've done genomics, transcriptomics, proteomics, microbiomics, epigenomics, and really importantly, they have clinical data for all of these samples. And they've made all of this publicly available. And if you want to access the raw data, you can go directly to the TCGA website. I will warn you that if you try to download the raw data, make sure you have a really good data connection because the files for this are absolutely massive. But what I'm going to do is I'm going to show you a really awesome website that was created that will allow people who don't code a lot like you and me to access the data and actually get a huge amount of information from it. So cBioPortal is a website that was made that is basically a portal into the TCGA data. So I should say it started as a portal into the TCGA data. Now it's actually a portal into the TCGA data and a whole lot of other data. So if I create a new data set, I can upload it onto cBioPortal and make it publicly available through their analysis tools. So many, many researchers are choosing to do that now. And so there's a ton of data that's on here. And again, you can upload your own data onto cBioPortal, and you do not have to make it public when you do this. You can choose to make it public, but you don't have to. There's a tab up here that says visualize your data. And so you can actually run in parallel an analysis with your data and the sequencing data that's publicly available. So we're going to go through and we're going to do, so basically what you do is you kind of choose a site of interest that you're interested in. And then it's going to show you what data sets are available for that particular site. And then we're going to choose a couple of them and we're going to do an analysis for specific genes that we're interested in. Okay, so one of the things that you can do is you can either choose, let's say you've got 5 genes that you're interested in, you can put those directly into the query box down here. The other thing you can do is you can choose a predefined data set. So let's say you're interested in looking at something like DNA repair. You can actually choose from the drop-down list of DNA repair genes, and it will just pull up a list of DNA repair genes, and then you can run the analysis on that. You can also pull down the DNA repair data set and then add a bunch of your own genes if you're interested in too. Keep in mind that if you put the name of a gene in and it gives you an error message, just look for alternative versions of the gene name because it's only going to accept it. It only considers one spelling to be the right spelling. So sometimes you may need to kind of figure out what it thinks is the right spelling. Once you kind of choose your data set and you hit the analysis button, it's going to pull up a whole lot of information. the first thing that you're going to see is going to be this oncoprint, and it's an overview of basically all of the data. So we chose a bunch of different genes. And what you can see here is each kind of vertical line down is the gene expression for all of the genes in one patient sample. So you can quickly see that, let's say, patients that have decreased expression of this ADAM10 gene oftentimes also have increased expression of this ARRDC gene. And so you can do this kind of really quick and dirty, just kind of visualization analysis. And the color coding is explained down here. Now, the other thing that you can do is you can take these data and you can actually make a heat map with them, which is also a great way to kind of visualize what genes are associated with what. So you can pull up different tracks, and there's a heat map track, and you can just add in all of your genes and say, add genes to heat map, and then it will generate this for you. And the nice thing about anything on cBioPortal is all of the visualizations that you see, you can download either the actual visualization as it is as an image file, and you can download the data behind it so that you can do analysis, let's say, on a program like Prism. So that's one of the beautiful things about it is you can always access the data behind there. One of the other really great things you can do is you can look at comparisons of gene expression, let's say in different subtypes. So here I look at different subsets of colorectal cancer, and you can see that I'm looking here at gene expression of this ADAM10 gene, and I can see that I've got generally on average higher expression in this MSI set than in this SYN set. Again, we're going to go over a live demo of this so that you can see it kind of in action. There is mutation information. So in the mutations tab up here, you can pull up any one of the genes that you're interested in that's in this data set, and you can look at kind of where the mutations are distributed in your gene of interest. And the nice thing is if you look at the table down here, it will tell you if they can predict whether it's going to be a missense mutation, or it's going to lead to a deletion, or it's going to be a silent mutation. So it can actually predict whether the mutation that you're interested in is going to be pathogenic or not. You can also look at co-expression. So let's say you've got a gene that you're interested in, and you want to know what is expressed along with this gene or what is not expressed with this gene. You can basically run an analysis on that. And one of the really nice things is that you can do comparisons. So let's say down here, I want to compare a group that has mutations in these genes and a group that does not have mutations in these genes. And what we've done here is kind of we've pulled up a volcano plot that has the differentially expressed genes in each of these sets. And then you can download any of the data that's actually behind any of these graphs. You can also do a survival analysis. So you can see there's a survival button here that you can look at. And you can see that in this particular case, if patients do not have mutations in the genes in this data set, they actually have a much better survival than the patients that have the altered genotypes. So it's a really, really powerful database or way of accessing the TCGA data. So let's flip over to cBioPortal so that we can actually do a live demo of it. And I'm going to choose bowel cancer just because I often do colon cancer. Well, I mostly do colon cancer research. I think you all know that. So you can see here that I've clicked on bowel, and I've pulled up all of the colon cancer data sets that are here. I'll also note that there is a pan-cancer studies tab up here. And this will actually, let's say you want to compare expression of genes or mutations of genes in different types of cancer. The pan-cancer data sets will allow you to do that. So you can choose that here. And the other thing is, in this pan-cancer data set, in addition to that, there's actually data sets of some cell lines. So if you use cell lines commonly, you want to know, does it have a mutation in a particular gene? You can actually use this software to do that analysis. So if we come back down here to bowel, you'll see I've got a whole bunch of data sets that I can choose from. On the right over here, you can see kind of basic information about the data sets. You can see some of them have a lot more samples than others. So this one's got 594 samples. This one has 22 samples. So you can kind of look at what is available. If you want more information, so each of these data sets is linked to the publication where it's been published. And it's also linked to kind of just an overview of the study. So let's say I want to do a quick overview of this particular study. I can click on that graph. And it will pull up just kind of general information about that particular study. So you can see that within that study, I've got different types of colon cancer, so colon, rectal, mucinous. I can see males and females. I can see, fraction of the genome that's altered. I can see the age of diagnosis. And you can actually go in and you can customize the tracks up here and actually choose like a whole bunch of different tracks that you want to look at. So let's go back to the actual innate analysis portal to do a gene analysis. We'll go back to bowel. And I'm going to choose the pan-cancer data analysis. So the samples that have all of those levels of annotation that I talked about before, the multi-omics, these are the ones that are going to say TCGA. The other data sets that are up here, like MSK, these may not have all of those different levels of data analysis that I talked about. That doesn't mean they're bad data sets. It just means that not every data set has everything available. But if you see one that is labeled TCGA, which for almost all these types of cancers, there is at least one TCGA data set, that's the one I would recommend as a first start, just because they're usually quite large, and also they have a really in-depth analysis of the genes that are present. So we can come down here to query by gene. And now you can see I can choose the different parameters that I want. So I can look at mutations, structural variations, copy number alterations. In my case, I want to look at expression. So I'm going to choose RNA. And I'm going to look at protein expression. The protein, I should say, is not there for all the data sets. They're still working on that one. So now I can choose a list of genes that I'm interested in, and I can enter them in down here. Or I can choose, yep.
For the mRNA expression, do you always do the one that's relative to the diploid samples?
Not necessarily. I'll actually often do relative to all samples.
Okay.
They don't necessarily always have the diploid samples in all of them. So,
yeah, cool.
And also for some of them, if they have normal available, you can look at the normal one. You can also, once you go into the analysis, you can actually also choose between these different data sets. So what you choose here is not the only thing you can look at. So I can go and I can either enter in the genes that I'm interested in, or I can come down here and you can see I've got different data sets that I can choose from. So let's say I'm going to choose, let's make Rosalind happy, and we'll look at the RB pathway. So these are pulling up genes that are associated with the RB pathway. If I wanted to add a different gene that I'm interested in, let's say APC, I can add that in right here. If I pull in, so let's say I'm going to put in a gene name that I know is wrong. So I want to look at CD8. It's going to tell me CD8 doesn't exist. Do I want to choose CD8A? So many of the time that you put in a gene name it doesn't like, it will suggest one for you. Most of the time that's right, so you just click on that. If ever it doesn't come up with a suggestion, it just tells you it's wrong, just quickly do a search for your gene and find out what the alternate names are. I've never looked for a gene and not found it here. I just have had to find the right spelling. Then you can submit query. And all of these data sets, all of these analyses are done live. So sometimes you have to sit for a minute or two. But generally, as you can see with that, it's pretty darn fast. So again, we've got kind of this general overview slide, and the number over here is giving you, like, within your sample set, this is telling us that 60% of these cancers have some kind of alteration in the CDKN2A gene. So it gives you a really good overview of that. If you want that heat map, so right now you can scroll down and you'll get to just kind of this legend. If you want the heat map, which I actually only found out after several years of using this, you can go to the tracks site here, and you can actually add in a whole bunch of different level analysis. If you wanted to add in a methylation track, you can do that. The heat map one is really nice because it gives you a really nice visual, and then you can just say add genes to heat map, and it will quickly kind of do the analysis for you. And now you can see I've got a heat map kind of right below where this original data set was. And if I want to download it, I can very quickly, where are we up here? You can see that there's a download tab right here. So you can download any of these visualizations or the data behind them. So that's one of the beautiful things about it. Oh, you've also got the download tab here. So you can download it in multiple formats. You can do a PDF. You can do the OncoPrinter, you can do SVG, you can do the patients. It will always give you several different choices. So we'll quickly look at kind of the plots, which is the one where you can actually look at expression analysis. So right now, it automatically kind of generates a graph for you first, and then you can go in and you can modify it. So right now, it kind of gives you two sections of things you can do. So you can choose what's on the horizontal axis and what's on the vertical axis. So right now, a lot of the time what I like to do is I like to look at clinical variables. So I can choose, let's say, a clinical attribute, and I can either look at something like a mutation count. I can look at the sample type that I have. Well, that's not a very exciting thing. Okay, so let's not choose sample type because that's super boring. Let's choose something interesting like, okay, so you can also type something in here. So we can look at pathology. What? Okay, so let's say we're going to look at the different subtypes. So you can see I've now got a graph with the different subtypes. And then I can choose. So this is my gene expression tab, and I've got mRNA. Again, I can change this, and I can change it to methylation. I can change it to microbes or protein. And then here, Lisa, this is where I can actually go in and I can change which of the RNA sets I'm comparing it to. So I can look at it relative to normal right now. And then I choose which gene that I'm interested in. So right now, CDK2, I can look at this particular set, and I can see that I've got way more expression in these particular subsets than I do in this one. And again, if I want to download the data, I've got my download button up here, and I can download either the image, the PDF, or the data. And the data will just be the expression values in a really simple tabular file that you can just open up in Excel or put into Prism. And it will allow you to do kind of the detailed analysis that you want. There's other resources up here. So you can choose, this is the survival one. We can go, so again, for all of these, you've got a bunch of different options here. So if you come onto this website, I really recommend you can click around and play around with it. I should also say that once you've done a search, if you create an account, you can save your search and you can come back to it and you can edit it whenever you want. That is one of the absolutely beautiful things about this is you don't do the search once and then lose it and have to do it again. You do the search and then you save it, and it will basically just give you like a URL and you come straight back to it so you can analyze it. You can save multiple different searches. It's really powerful in that way. So if you want to do survival, I can come to survival over here. And you can see that here it's showing me altered versus unaltered. And what I found rather annoying about this is it looks at, normally what you're interested in is you're looking at like one particular gene, and I'm like, if I have a mutation in this, does it affect survival? The default survival on this is it looks at samples that have alterations in everything versus samples that have alterations in none of the genes, which is mostly not helpful. So oftentimes what I will do is I will come in and I will just kind of, oops, now it's taking these out. Okay, so hang on. So if I deselect all, now I can go in and I can choose one of the particular genes that I'm interested in, and I can look at survival relative to that particular gene. I can choose two, and then I can look at survival of samples with a mutation in one of these genes versus sample with a mutation in the other. So it's really flexible, and you can kind of change what you're looking at in terms of the different parameters and different combinations. You can change the visualization. There's some options up here to be able to do that. Again, you can kind of zoom in here. Oh, and I should clarify. So this is overall survival. For some of them, you'll actually be able to scroll down and there'll be like disease-specific survival and things like that. Again, not all of the data is available for all of them. So some of the ones you'll see, sometimes you see these little warnings that some of the patient data doesn't correspond, but it will always warn you about that. So you at least have that information. Now, one of the other really important things is you can look at, you can download basically any of these individual pieces of data that you want. So let's say I want to download all of the copy number alterations. I can just download it in a tab delimited format, which is easy to open it in Excel or any editing platform. You can access the RNA data. You can access pretty much all of these levels of data in basically a really accessible form that you can then put into, you can put it into R if you use R, but you can also then put it into just Prism. Let's say you want to use some of these data in a paper and you want this figure to look like all of your other figures, then you can kind of download the data and analyze it with Prism with your specific parameters and make it look kind of uniform. So it gives you a hugely powerful set of data that you can look at. And you can scroll down and you can see that there's all kinds of information here. And each of these individual samples is a patient. So you can actually kind of, if you want information on a specific patient, so let's say I want to look at this specific, that was the study, not the patient. Let's just scroll back. So let's say I actually wanted to look at the specific patient. I can actually go here and I can click on, let's say, patient ID, and you can get actually a breakdown of what that patient's information is. And they'll tell you kind of like really detailed analysis, like this is the treatment that the patient received. These are the mutations in the tumors. You can look up here. This is one of the really interesting things. Oftentimes they'll actually have a tissue image. So they'll actually take a picture of what the tissue looks like. It looks like there may not be one for this particular sample. Of course not, because that's the one I chose. It actually shows you the pathology report. So it'll tell you exactly what the sample was. You know, there was a tumor that was measuring 1 by 1 by 0.3 centimeters. So you can get all this data. And again, most of the time you don't need this data, you just need the other data, but it can be very, very useful to kind of have access to this if you want to dig into it a little bit more. All right, so let's move on to the next resource that I'd like to discuss. And this is going to be the UCSC Xena Cancer Browser. And this is similar to TCGA, but what's different about it is it will actually allow you to make comparisons between different data sets. And that's one of the things that I think is particularly powerful about it. It also makes really great visualizations. It will look at gene expression and clinical parameters. It doesn't have like the epigenomics and the things like that. But the data can also really be easily sorted and filtered, and you can choose particular cutoff levels for expressions, which is what makes this tool a little bit different from some of the others. So you can see once you get into the portal, you basically choose a disease type, and you can choose, here I'm choosing one particular study, but I can choose multiple. Then you can kind of choose a first variable, which could be a gene or a particular phenotype. And again, we're going to do a live demo of this in a minute. So in this particular case, I've chosen a colon cancer data set. I've said I want to look at genomics and I want to look at expression of this protein APC. And then again, I can look at gene expression, copy number, somatic mutation. And if I click the advanced, I can choose a whole bunch of additional parameters. What you'll end up with is, and I'll show you how we're going to get there, an image that's going to look like this. So I've started out, I told you we had APC in the last one. Now I've also added a second gene that I'm interested in. I've added KRAS, and I can add in as many variables as I want here. And I can actually see how each of them is related to each other. So again, each line like this corresponds to one individual patient. And I can go in and I can recluster these however way that I want. So I can, again, put multiple parameters. I can choose a clinical parameter. So phenotype, I can look at the pathology, which is what we're going to look at in a minute. And then up here, I can choose particular cutoffs. So let's say instead of looking at all the KRAS, I want to look at KRAS with only one particular mutation. I can enter in that mutation information here. Or I can enter in, I want to look at samples that only have expression index above 5. So that's one of the things that this tool will allow you to do that TCGA will not. And again, you can download all of these data, you can analyze right in the window, and you can graph the data. So we're going to do a quick demo of this in action. So this is the Xena browser. You do have to create an account for this. It is completely free. They will not spam you. I have not received a bunch of spam. You can ask my students. I always warn them when I'm going to get a bunch of spam from a website. You will not from this particular one. So if we start and we jump right in. All of these sites, by the way, also have fantastic tutorials. So I definitely recommend taking a look at those tutorials. So over here, I can choose, if I know the study I want to use, I can start typing in information here. You can see that this contains sets from different papers. So this is going to be the citations for the papers. I can say, no, let's say I want you to help me select the study. So let's say I'm interested in breast cancer. I can search breast and I can say I want to look at breast cancer. And then if I click here, it's going to pull up a bunch of different potential data sets that are related to breast cancer. Somehow I'm also getting samples with cervical cancer. Anyways, so we can choose, let's say, a breast cancer data set. Let's say, I'm just going to choose a random one. So let's say breast cancer from this. Let's choose the TCGA one just because I know. So I can choose now. So now that I've set this, I can say this is my first variable. And now I can select my second variable. So I can either make this a gene that I'm interested in, or I can make it some kind of phenotype. So let's say I'm interested in looking at something like survival, or let's say age at diagnosis. So I can say age at initial diagnosis, and I can add that as my second variable. It always default sorts by this. I can also do, let's say I want to do phenotype again, and I want to choose pathology. So let's say I want to look at what the pathologic stage is. It's going to let me do it. I'm going to go to advanced. I'm going to say pathology. Here we go. So let's say I want to look at whether the patients have lymph node metastases. I can come down and I can choose this, and I can add it. And now I can add, so if you want to add additional ones, there's this really small little button here that you just click on it, and now I can go in and I can add additional things. So now I can add in, let's say, a gene that I'm interested in. So with breast cancer, everybody thinks it's the BRCA gene. So I can say I want BRCA. And then I can say, do I want to look at gene expression, copy number, somatic mutations? I can look at more things. Again, if I go into the advanced, I can look at mRNA expression. So there's a lot of different things that you can choose from. We're going to go with just gene expression to make life simple, and I can add this. And so now keep in mind that I can keep going through and I can add like 10 different genes here. And then I can go on to any one of these and I can go ahead and sort them if I want. So let's say I want to sort by this particular one. I can then choose to sort by this one. So I can say reverse sort, and it's going to sort based on that. Now, the interesting things comes up to when I want to do the actual comparisons. So if I come now over here, this is where I can download the data. So let's say I don't want to download all of the expression data. I just want to download expression data for some things with the survival data or with the age at diagnosis. Or I can do an analysis where I go view as chart. And this is where I can actually do kind of a comparison of subgroups. I can make a scatter plot. I can look at columns. So let's say I choose compare subgroups. And I say, okay, I want to look at end stage. So whether or not we have a node. Hang on a second. Show data from age at diagnosis. Okay. And then I can choose one of the parameters that I have down here. So we're going to look at N. And let's say I'm going to look at a violin plot because I like violins better than box plots. You click OK, and it will generate a violin plot for you. So on the left here, I've got age at diagnosis. On the right here, I've got end stage. So I have patients that have no lymph node metastases and patients that have lymph node metastases. And it actually will do the statistics test for you. And as you can see here, there's no statistical difference between this. So you can then actually do, again, you can download the data specifically associated with this graph, and you can modify. You can see when you hover over it, you actually get the information specifically related to the set. So you can kind of layer in as many different genes as you want and actually run a bunch of different comparisons. So again, this is a really powerful software tool. Okay, so if we come back to our PowerPoint, okay, so this is an example of kind of using that data set. Now the UCSC genome browser, so UCSC, by the way, is University of California, Santa Cruz. They're the ones who put together the Xena browser we just looked at, and this is the genome browser. And for any of you doing genomics as opposed to looking at expression data, I thought I would just kind of very quickly show you that this set is available. A lot of us will look at NCBI if we look at gene information, but the UCSC genome browser is actually extremely powerful. You can do a gene level analysis in many different species. And the nice thing about it is you can look at features you can't look at in the other data sets. So you can look at things like promoter methylation, regulatory regions, introns and exons, the SNPs, if you're interested in SNPs. You can look at transcripts, repeat sequences. And a lot of us are doing CRISPR nowadays. You can actually find CRISPR sites on here. And it's nice because it gives you a really cool visualization. So if you choose a particular genome, you can enter in a gene up here, and it will pull up a page that looks kind of like this. And I know this looks like a mess, but if you actually take the time to look at it, it's actually quite organized. So you've got your chromosome here, you've got your RNA, sorry, your gene here, and you can see introns and exons are very clearly marked. You can look at the RNA values here. You can look at gene expression. So if you click on this one, you can actually see what tissues it's expressed in. You can look at methylation. So you can actually see, so this is the methylation regions associated with your genes. If you're interested in looking at promoter methylation, you can scroll a little bit to the left and you can actually see what's methylated before the start. You can look at, so this is going to be common SNPs that are present. And for each of these, you can click on them and get additional information. In my case, I'm interested in repeat sequences, so I look at repeat sequences. But you can get a lot of information and you can scroll down and there's a whole lot of different tracks that you can enable here. So this isn't particularly patient data, but I thought I would point it out because I think it's a really powerful tool that a lot of people don't know about. All right. So gene ontology is the next thing we're going to look at. And this is something that a lot of people do. So let's say you do sequencing. The gene ontology is going to give you a way of looking at, let's say you have a huge list of genes and you want to know, like, are these genes associated with specific functions or signaling pathways? It's the gene ontology analysis that you're going to want to be able to do. Gene ontology, so this is the major website. And I'm going to show you how to do an analysis of a single list of genes. So let's say you've downloaded a data set and you've identified that, like, in luminal A breast cancers, I've got these particular genes that are enriched above, let's say, these genes that are expressed in triple negative breast cancer. You can take that list and you can put it into here, and you can actually do an analysis and get an idea of the functional things that are associated with the luminal A cancers as opposed to the breast cancers. So this is an example of, when you download a data set from the Geo database and it doesn't have that analysis button for you, this is a software that you can go ahead and use, and you can actually analyze those data yourself in a very comprehensive way, and a very simple way too. So basically you put your gene list. Once you click on that button, it actually brings you to the analysis site, which is called Panther. It will allow you to look at gene expression signatures and sets of genes. And it will allow you to look at, so you can only look at one list of genes at a time. So you can't compare different gene lists. I'm going to show you how to do that one in a minute. But you can look at an individual list of genes and you can see what types of functions are associated with this. And you can also see, let's say I have this particular gene set, am I overrepresented in specific functions, which means that these functions are specifically in my particular type of cancer. The nice thing, again, is it will do all of the statistics for you. So that's one of the beautiful things about this. So you would basically enter in your list of genes here, just copy and paste, or you can have an Excel file and upload it here. Just choose what organism you have. For most of us, it's going to be mouse or human. And then you can choose what you want to look at. You can look at a functional classification, statistical overrepresentation, or an enrichment test. And you can click submit, and then it will do kind of the analysis for you. If ever you don't want to just do like a random search, but you actually want to find gene signatures that are associated with specific functions, where you can find that is a website called MSigDB. It stands for Molecular Signatures Database. And this is basically where all of the curated gene sets are to be found. So this is where you can find genes associated with apoptosis. You can find a gene set that's associated with metastasis. So it makes it very, very easy to kind of access a big data set, or access genes that are associated with a particular function. And then you can actually use that and run comparisons with your own thing. All right, so we're going to go on. So I'm going to quickly kind of show you that on the website. So this is going to be, here we go. So this is the gene ontology resource. And you can see what I've done here is I've just kind of pasted in a list of genes. I did it in advance because I knew I was going to be crunched for time. Oops, here we go. And you can see I can choose what I want to look at. So let's say I want to look at molecular functions. I've got mouse, and I can go and I can launch. And this is going to basically pull up a list of functions that are specifically associated with my genes of interest. So you can see here, so it's done an analysis. Oops, I'm cutting off here. There we go. You can see that it's actually showing me that of that list of genes that I pulled up, the functions that are associated. So we've got an enrichment in things like protease binding, and ubiquitin-like binding. And again, all of the statistics are over here. showing me what is significant and what is not significant, kind of comparing against gene signatures. So it's a really powerful site. Now, again, this will not allow you to do comparisons. Now, the site you want to use if you want to do comparisons, and I know we're running at time, but I'm going to take an extra 10 minutes of your time for anybody who's willing to listen. I will not go above that, is Metascape. This is a phenomenal website. And I have seen more and more papers actually say that they did their analysis in Metascape. And like really, I've seen papers in Nature and Science that are saying, I did my analysis in Metascape. So don't be shy to use this. There's a lot of people in high profile labs who are using these tools. There is no shame in using them. So Metascape is awesome. It gives a comprehensive gene expression analysis. It includes comparisons of samples. It includes statistical analysis. The list, what you want to input is basically a list of genes that are differentially expressed. So let's say you've pulled your data set from the websites and you found that, we've got 20 genes that are upregulated in luminal A breast cancers and 45 genes that are upregulated in triple negative breast cancer. And you want to know, like, functionally, what is the difference between them? Are there overlaps between them? You may not have done a differential analysis ahead of time. You may just have a list of genes you want to query. So it's easy to do that. And the really nice thing is all of this is going to generate a nice little exportable report for you. You basically just click on the export analysis, and it's going to basically export it for you, which is absolutely beautiful. And then you can use it however you want. So this is an example of what the data is going to look like. I'm going to go through to the live test of it just so you can see it in action. So this again is Metascape. I have put in just a simple Excel file that has 2 columns. One of them is genes that are expressed in SIN cancers. One of them are genes that are expressed in MSI cancers. I took these from a public data set that I exported, and I basically just downloaded those data in an Excel format, and I just uploaded them. I created an Excel file, and I just uploaded it into here. I did choose, so if you have multiple genes, you want to make sure you choose this multiple list option. You want to tell it what species you have. So in my particular case, I had human Homo sapiens. This was the analysis I had. And then we're going to click, you don't need to do the custom analysis. You can do the express analysis, and that is just fine. So I'm going to warn you, once you press this button, it takes a bit of time. So I'm pressing it now, and you're going to see it's going to like run through the analysis. So don't be, if you press something and nothing happens immediately, that's normal. I'm going to quickly swap over to this tab where it finished the analysis before. And what you can see here is I've got, there we go, zoom in so we can see this properly. So you can see, first of all, up here, I can export either the Excel sheet of the genes that I put in. I can download everything as a PowerPoint, right? It will take all of this data that I'm going to show you and put it in a PowerPoint, and you can download it all as a zip file. So what we have here is we have a heat map. And this is showing me genes that are expressed. So I shouldn't say genes. This is gene ontogeny. So these are the pathways. So these are the molecular signatures that are associated with the MSI cancers versus the SIN. And so I get a really clear analysis and a really nice visualization of what is the same between my different conditions versus different. It only puts 20 in the default one, but you can see here, if you want the one that's got 100, you basically just click on this button here and you get the one that has 100. Again, you can download the individual images, or you can do the export all as compressed, and you get all of this. It will give you an idea of, so it's giving you here a color coding. So keep in mind that this color coding is going to apply to the next graphs that we're looking at. The really nice thing about it is it also gives you above all of these, above everything, it tells you what it did to get from the step before to the step after. So everything is very transparent and it's very explainable, and it has references for everything that it uses. So you can always trace things back. So it's done and it's gone a gene annotation. So it's just basically right now it's telling you the process that it went through. So it did functional like classification of all of these things. And now here we scroll down and we can actually see. So what's in the red is MSI, what's in the blue is SIN. And you can see that here it's looking at whether these two processes, so this particular process, so positive regulation of cell death, what this is telling me is that they're very differentially expressed between my 2 data sets. And you can see that it's gone through and it's basically pulled all of the relevant data set, or all of the relevant functional signatures. It's pulled all of these and it's showing you the statistics for them. And again, if you do the export analysis, it will send it all with all of the details and all of the genes for each of these. All of those will be given to you in a handy dandy Excel file. Again, now we can look at the actual pathway enrichment. So it gives you these maps, and it will show you, if you keep scrolling down, it gives you kind of a holistic one. So this is like all of the genes. Again, SYN or MSI is color coded here. So you can put in 10 different types of categories and do your analysis that way. And then it will do the breakdown of each of these. And all of these are clickable and interactive. So you can actually go into each of these and actually look at them. So it gives you a whole swath of data. The other really nice thing that it does, so I'm just scrolling through, these are all the signaling pathways. It does a quality control check, and it actually looks, the quality control check is not super interesting, but it does look at whether you've got genes that have been associated with like previous tissue signatures. What's a lot more interesting is this one down here. It actually shows you if the pathways that are associated with your different cancers are regulated by specific transcription factors. So this actually allows you to know like which transcription factors you would really want to look into for your analysis kind of after the fact, or if you wanted to dig into them later. And again, all of the statistics are here, and all of the references are present at the bottom. So again, if you click on that export kind of compressed file at the end or at the home, there we go. This all-in-one zip file, it exports all of this for you as a beautiful compressed file with all the images, all the raw data, all the analyzed data. So, and all the statistics done for you. And again, you can take that and you can put it into your own visualization tool if you want. So super awesome software. And again, you can do your analysis this way, and you can say in your methods section, I did my analysis with Metascape, and cite the paper, and you are good to go, because all of that is very transparent and their algorithms are very transparent. Okay. So I'm just now going to very quickly talk about a couple of the, so additional gene resource sites that are available. This one's going to be quick. So Gene Pattern is a way of basically, so what Gene Pattern does is it takes like all of the coding in R, and it allows you to really do a custom analysis without having to know the coding. You have to bring a data set. You can't just download one. So if you, let's say, download a public data set, you can bring it into Gene Pattern. It's definitely not for somebody who doesn't want to invest time in learning how to use it. So you can go in and you can choose modules that you want to look at, and you can actually choose specific R programs that you want to run the analysis. So you have to have some familiarity with what these programs do. It will do all the heavy lifting for you once you've chosen what your pipeline is, but you actually have to know enough to choose that pipeline. So a good resource for somebody who's a little bit more advanced. So some other genomics resources that I just want to quickly touch on, and I'm going to do, I'm not going to show you how any of these work, I'm just going to tell you they're available. A lot of the sequencing data that you download is like, it's not single cell sequencing, it's total RNA-seq. And so you don't get a breakdown of the cell types. There is a program called CyberSort, again, which you just run online. And if you put in expression, so let's say you've downloaded your public data set, you basically paste that into CyberSort, and it will actually tell you, oh, you've got gene signatures associated with fibroblasts. Let's say 40% strength fibroblasts, 40%, 20% CD8 T cells, 30% cancer cells. And so it will give you, it's an estimate, and it's an estimate based on gene expression signatures from different cell types. But it's really well accepted. I'm sure you have likely seen CyberSort in some of the papers that you've read. And so it's a good way of taking kind of whole tissue expression analysis and actually getting an idea of what cell types are there. Estimate is similar. So by the way, for all of these websites, I've put the link at the bottom here, and I will share a copy of this presentation with Krina. And if you would like a copy of it, you can either email me or them. Estimate is another really good one. This one is really specifically for stromal and immune cells. It gives you a little bit, it breaks down the immune cells into a lot more sophisticated subsets for anybody who's interested. The ICGC data portal, it's very similar to the other cancer portals that I've talked about, but it has really particularly good visualizations. The reason I'm showing you multiple of these, by the way, is because different people find them, will find one more user-friendly than another. So definitely don't be shy to explore these. So all of what we've talked about is kind of gene level expression, and a lot of people will tell you we want protein. And so there's just many fewer resources available. I'll be completely honest with you, there's many fewer resources available. That's starting to change. One of the main sites you can use if you want to look at protein is called the Human Protein Atlas. Website is down here. And this will basically just allow you to put in a particular protein that you're interested in and get an idea of what its expression level is in tissues. But there are these two boxes down here for pathology and disease. And you can actually go into those and actually look at whether at the protein level, your gene tends to be up or down regulated in different cancer types, and whether there's an association with survival. So it's kind of basic, but it's as good as we've got for a lot of protein level analysis right now. Now, the other thing I want to just briefly touch on is single cell sequencing. Some of the data sets, so in the Geo database, you can download single cell sequencing from there. You can't do the analysis for single cell sequencing there because the analysis for single cell sequencing is quite a bit more challenging than just for total cell sequencing. But there are two websites that have recently been created that let you do like analysis of publicly available single cell sequencing data sets. You do not have to bring the data set with you. These sites are directly connected to, let's say, the single cell sequencing data sets that are already publicly available. So these sites have to have already chosen a data set, but most of them that I looked at have at least some single cell sequencing for a lot of different cancer types. So you can go in and you can do like your UMAP clustering and things like that, and you can correlate it with pathology. It's a little bit more advanced than kind of what we did today, but if you go onto these websites, you'll be able to figure it out without too much trouble. And again, there's a lot of really good tutorials on all of these. The other good one for single cell sequencing is called Zenodo. I prefer this one, but they're both good. For anybody who wants just like general cancer statistics, what are the rates of breast cancer in Canada? I really encourage you to go on to cancer.ca, the cancer statistics website. They release a report every year on like the current status of cancer, and they release a lot of really valuable data and really granular too. Like you can look at the rates of luminal A breast cancer in Alberta very specifically. So something that you may be interested in looking at. Again, there's no genomics data, but I just thought I would mention it. If you want to look at it in the States, this is the equivalent, the SEER statistics by Cancer Control Government Alberta. So summary. Okay, I went 2 minutes over. So there's a lot of publicly available data out there that you can use to enhance your research. And there's a lot of publicly available resources that mean you don't actually have to be an awesome coder to use this data. And I think that we really should use the data, again, because it's available. It makes our research stronger, and it gives credit to the people who have done the work. So I did want to acknowledge a couple of acknowledgements. So number one, all the patients who've donated their samples to these databases. It doesn't seem like a lot because most people just want their cancer tissue gone, but it does take a lot for the patients to kind of do this. All the researchers who did the analysis, this has taken a lot of time and money, but they chose to do it and they've made it publicly available. And also the researchers who build the software tools to make it accessible to people like you and me who are not like super strong coders. If some of you are super strong coders, that's great. But for the rest of us, this is what's available. So if anybody has questions, I'm happy to stick around and answer them. I hope that was helpful in some capacity or other. If anybody has questions in the future, please go ahead and just reach out to me.